Welcome back to The Deep Dive, the show where we grab a stack of research, articles, and notes, and really distill it all into those essential nuggets of knowledge or insight you need to be truly well-informed. Today, we're plunging into a topic that quite literally affects every single one of us, every single day. The invisible world of environmental chemicals and their profound, often unseen impact on human health.
Indeed. It's a vast and complex landscape. While mammals have evolved these incredible, sophisticated mechanisms over millions of years to protect us from many chemicals found in our natural environment.
Right, like detox pathways.
Exactly. But the sheer volume, the diversity, and the novelty of agents introduced, particularly since the Industrial Revolution, they present wholly unique challenges that our bodies are, frankly, still trying to grapple with. Our mission today is to embark on a journey with you to deeply understand how chemicals in our environment, our homes, our workplaces can adversely affect our health. We'll be covering fundamental principles, diving into the specific toxicants, and exploring the cutting-edge science of how our bodies react and adapt, or sometimes fail to adapt.
That's right. You've provided us with a truly fascinating collection of sources, including deep insights from a foundational text like GOODMAN and GILMAN's The Pharmacology. Our goal by the end of this deep dive is to help you master this complex information, just like the elite professionals in this field. We're going to unpack everything from how environmental risks are assessed to the nitty-gritty of specific metal toxicities, the subtle but powerful influence of endocrine disruptors, the insidious effects of immunotoxicants, and even the surprising critical role of our own gut microbiome. So consider this your comprehensive guide to understanding environmental health. Let's get started. 1. The silent threats. Understanding environmental and occupational toxicology. It's almost like our bodies possess a superpower, isn't it? These incredible built-in defenses against foreign chemicals, or xenobiotics as they're called, thanks to complex transport systems that move things around and metabolic mechanisms that break them down. But clearly, that adaptation has its limits, especially with how dramatically our world has changed.
Absolutely. The very evolution of these protective systems, our liver's detoxification enzymes, our kidneys' filtration capabilities, it highlights the constant exposure to chemicals throughout history. For millennia, our ancestors encountered chemicals in food and water, in the air, but these were largely natural, predictable compounds. Our bodies developed sophisticated ways to process, neutralize, and eliminate them. However, as you alluded to, the Industrial Revolution fundamentally shifted this landscape. Suddenly, we introduced countless chemicals that were previously rare or entirely absent from the natural environment: synthetic compounds, industrial byproducts, new materials. These novel compounds in unprecedented volumes are what truly challenged our evolved defenses. This dramatic and rapid change spurred the creation of dedicated scientific disciplines. Environmental toxicology, which is the study of how chemicals in our broader environment adversely affect human health. And occupational toxicology, which focuses specifically on chemical exposures within workplaces, understanding the unique risks faced by workers. These are not minor concerns. They involve significant situations where environmental agents cause profound, often chronic, toxicity. We're talking about substances that can silently chip away at our health over years, even decades, without immediate, obvious symptoms.
And we're not talking about a quick drug trial here, are we? Assessing these environmental risks poses unique, almost daunting challenges compared to, say, developing a new pharmaceutical.
Exactly. The fundamental difference lies in intent and control, which profoundly impacts how we assess risk. Unlike a drug, which is given for a specific disease with an intended therapeutic benefit that we hope outweighs the known risks, environmental toxicants are generally only harmful. There's no inherent benefit to being exposed to lead or asbestos or certain industrial chemicals.
Right, no upside.
Precisely. Furthermore, exposures to these environmental toxicants are almost always involuntary. You don't choose to breathe polluted air or drink contaminated water in the same way you choose to take a medication for a specific ailment. This involuntary nature profoundly impacts how risks are perceived and accepted by the public. As a result, the focus of environmental risk assessment is critically on the low end of the dose-response curve. We're trying to understand the effects at concentrations that might seem negligible, but over chronic involuntary exposure can be highly impactful. This often requires very long-term chronic exposure experiments, which are inherently complex, expensive, and difficult to extrapolate to humans. There's considerable uncertainty about the precise severity of effects at these very low doses, and understandably, people are much less willing to accept these involuntary risks compared to a calculated risk taken for a known therapeutic benefit. The margin of error, or rather, the margin of safety, needs to be much, much larger. 2. The science of risk. how we measure and manage environmental threats.
So if these are silent, involuntary threats, how do we even begin to predict the effects of these exposures? It feels like trying to see the invisible, especially when the health impacts might not show up for years or even decades. How do scientists piece together this complex puzzle?
It's a multifaceted approach, one that truly integrates two complementary scientific powerhouses, epidemiology and toxicology. They offer different yet equally crucial lenses to view the same problem. Epidemiologists act like public health detectives. They monitor real-world health effects in large human populations and use statistical analysis to associate observed health issues with specific environmental stressors.
Okay, so they're looking for patterns in people.
Exactly. They might notice a higher incidence of a particular disease in a certain community and then investigate if there's a common environmental factor at play. Toxicologists, on the other hand, perform controlled laboratory studies, typically using animal models or cell cultures, to meticulously examine the potential mechanisms of a chemical's toxicity and predict its potential harm to humans. They're trying to understand the how at a molecular and cellular level. The data from both fields are absolutely crucial, and they necessitate integration to inform comprehensive environmental risk assessment, which then helps develop real-world management approaches like laws and regulations, ultimately aiming to limit our exposure below what's considered safe.
Okay, let's unpack this detective work first. How do epidemiologists actually track health effects in real people, with all the variables out there? Human life is so messy and full of confounding factors like diet, lifestyle, genetics. How do they ever get a clear signal?
That's precisely the challenge, and it's why they employ a variety of sophisticated study designs, each with its own strengths and weaknesses. It's like having a different kind of magnifying glass or a specific tool for different parts of the puzzle, depending on the question they're trying to answer.
Reading the human story.
It sounds like a massive detective story. What are some of the main ways they piece together these human stories, especially when they can't control all the variables, like in a lab?
Right. They use a variety of study designs, as you said, each suited for different investigative questions and different stages of inquiry.
Okay.
First, you have ecological studies. These studies look at correlations between the frequencies of exposures and health outcomes across different geographical regions or large populations.
Like comparing cities.
Exactly. They might look at average air pollution levels in different cities and compare them to the average incidence of respiratory diseases in those same cities. They're relatively inexpensive and great for generating initial hypotheses, pointing to areas where more focused research is needed. But, this is a big but, they are highly susceptible to confounders, other unmeasured factors that might explain the correlation. And crucially, they cannot establish direct causality.
So correlation isn't causation.
Precisely. Just because two things happen in the same place at the same time doesn't mean one caused the other. For instance, seeing higher cancer rates in one region with high pollution doesn't automatically mean the pollution caused it. Maybe that region also has higher smoking rates, or different diets, or less access to healthcare.
Got it. What else?
Then there are cross-sectional studies. These examine the prevalence of both exposures and outcomes at a single point in time. Think of surveying a population today to see who has been exposed to a certain chemical and who currently has a particular health condition.
A snapshot in time.
Exactly. They're relatively inexpensive for finding associations, but they don't show a temporal relationship. You can't tell if the exposure happened before the outcome, or vice versa. So again, they can't establish causality and are prone to various biases. For example, a health outcome might cause someone to avoid an exposure, skewing the data.
Makes sense.
Next up are case control studies. Here, researchers start with a group of individuals who are already affected by a disease, the cases. They then carefully match them to unaffected individuals, the controls, who are similar in known confounders like age, sex, etc.
So they work backwards from the disease.
Right. Questionnaires are often used to assess past exposures. This method is relatively inexpensive and particularly good for examining rare outcomes because the endpoint, the disease, is already known. You don't need to wait years. However, they rely heavily on potentially unreliable memories of past exposures. People might not remember accurately, or their recall might be biased by their current illness.
Okay, so memory can be tricky. What's considered more robust?
That would be cohort studies. These are considered much more robust for establishing causality. They measure exposures in a large group of people first, for instance, tracking workers in a certain industry versus office workers.
So you know the exposure before the outcome.
Exactly. Then, they follow that group over a long period, sometimes decades, to measure subsequent health outcomes. They're less susceptible to bias than case control studies because of that clear temporal relationship. This makes them much better at establishing causality. The trade-off. They are expensive, incredibly time-consuming, and especially challenging for rare outcomes. You need a very large study population to get enough cases for statistical significance.
Wow, decades. That's a huge commitment.
It is. And finally, clinical trials. While ethically impossible to intentionally expose people to harm to measure toxicant effects,
Obviously.
clinical trials are used to examine the effectiveness of interventional strategies. For example, testing if providing filtered water reduces lead poisoning symptoms, or if a dietary change mitigates effects of another exposure. So they test solutions, not the initial harm.
Given all those challenges, especially the inherent difficulties of attributing toxicity to one chemical entity when the exposure includes myriad other chemicals, because, in the real world, we're exposed to a cocktail, right? How do scientists get a clear signal? Is there a way to measure directly inside the body?
That's a perceptive question, and it's precisely where biomarkers come in as a really powerful tool. Because it's so difficult to assess real-world human exposures, which are often to complex mixtures, and because effects can take a very long time to manifest, epidemiologists often rely on these biological indicators. Biomarkers are measurable changes in biological systems in blood, urine, hair, tissue that provide objective information about exposure, effect, or an individual's susceptibility.
Oh, okay.
The ideal biomarker aims to be specific, detecting changes only in affected individuals, and sensitive, detecting changes in all affected individuals. They offer a more direct internal measurement of what's happening.
So what kinds are there?
Well, we can break them down into three main types. First, biomarkers of exposure. These tell us about the dose or duration of an exposure. We can measure recent exposures by looking at the chemical itself or its breakdown product, its metabolite, in blood or urine.
Like a drug test?
Sort of, yes.
Yeah.
For tracking exposures over longer periods, months even, hair and toenails can provide insights as chemicals accumulate there. And a really striking example is using X-ray fluorescence to measure bone lead levels. This can estimate lifetime exposure to lead a direct window into decades of exposure history because lead stays in bone for so long.
Wow, decades in your bone.
Exactly. Second, biomarkers of toxicity. These measure toxic effects at a subclinical level, meaning before overt symptoms appear. This is crucial for early detection and intervention.
Catching it early.
Right. Examples include measuring specific liver enzymes in serum, like ALT or AST, if a chemical targets the liver. Changes in urine contents, like protein or glucose, can indicate early kidney damage. And specialized neurological or cognitive function exams can detect subtle brain effects.
Before they become obvious problems.
Precisely. And third, biomarkers of susceptibility. These predict which individuals are more likely to develop toxicity in response to a given chemical, because we're not all the same, right? Genetics play a huge role. A key example is identifying single nucleotide polymorphisms, or SNPs, common genetic variations in genes for metabolizing enzymes. These variations can affect how quickly your body activates a toxicant or detoxifies it. Someone with a slow metabolizer SNP for a detox enzyme might be much more susceptible.
So your genes can make you more or less vulnerable.
Exactly. And what's truly fascinating is that some biomarkers can give you information across categories. For instance, measuring N7-guanine adducts from aflatoxin B1 in urine tells us both about exposure, someone encountered it, and about DNA damage, which is a direct toxic effect. It's like a 2-in-1 internal signature of risk.
That's incredible. Our bodies are literally recording the story of our exposures if we know how to read them. So if epidemiologists are reading the human story, what about the lab side? How do toxicologists approach this given they can't ethically experiment on humans?
Lab studies and predictive power.
Right. Toxicologists use carefully controlled model systems, primarily experimental animals like rats and mice, and increasingly cell cultures to meticulously examine chemical toxicity and predict human effects. The goal is to isolate the effect of a single chemical under controlled conditions. They typically test single chemicals over a large dose range, often at concentrations significantly higher than what might be environmentally relevant, and over shorter time frames like months or a couple of years.
Higher doses to see an effect faster.
Essentially, yes, it's done to achieve robust toxic outcomes and get statistical significance within a reasonable experimental period. You need a clear effect to study its mechanism.
But I imagine that creates its own set of uncertainties when trying to apply it back to us, to real-world scenarios, low doses over a lifetime, maybe mixtures.
Exactly. That extrapolation is a major challenge. There's often considerable uncertainty about the effects of very low doses or combinations of chemicals, especially when real-world human exposures span decades. To try and bridge that gap, toxicologists deeply study the mechanisms involved in a chemical's toxic effects. They aim to predict if that same molecular or cellular mechanism would occur in humans. If the mechanism is conserved across species, the chemical hits the same target in a human cell as a mouse cell, then the animal data becomes much more predictive.
Okay, so understanding how it works is key. What are the key findings from these animal studies that regulators actually use? The numbers.
Right.
To predict toxic effects and set guidelines, toxicologists perform standardized studies. These include subchronic studies, maybe 3 months for rodents, and chronic studies, typically 2 years for rodents, often in at least 2 different species. Doses are chosen carefully based on initial tests. They aim for one concentration with no significant effect at baseline, then one causing statistically significant toxicity at the low end of the dose-response curve, and maybe one or more with moderate to high toxicity. These studies yield 2 crucial numbers. The NOAEL, no observed adverse effect level.
Okay.
This is the highest dose used in the study that did not result in a statistically significant increase in negative health outcomes compared to controls. Yeah. It's the highest dose where they saw nothing bad happen statistically.
Got it. And the other?
The LOAEL, lowest observed adverse effect level. This is the lowest dose that did result in a significant increase in toxicity. It's to the point where a harmful effect was first observed. NOAEL and LOAEL. Okay, so what does this all mean for developing regulations? How do agencies like the EPA use these numbers to actually protect us from those low-level long-term exposures?
The NOAEL is a critical starting point for regulators. It's used to derive a reference dose, RFD, which is basically an estimate of a daily exposure level that's likely to be without significant risk over a lifetime. To get the RFD, the NOAEL is divided by what we call uncertainty factors or safety factors, typically factors of 10 for each source of uncertainty.
Factors of 10. Why 10?
It's a standard practice. The most common factors account for interspecies variability, the differences between animals and humans that gets a 10x factor. And inter-individual variability is the differences among people. Because some are more sensitive, that's another 10x factor. So typically, the RFD might be the NOAEL divided by 100, 10 times 10. Other factors might be added if the data isn't perfect, like if there's no chronic study data, maybe another 10x factor. This use of factors of 10 is a direct application of the precautionary principle. Essentially, you assume a worst-case scenario for each unknown, erring on the side of caution to build in a very wide margin of safety.
That makes sense, building in safety buffers. But what about those very low concentrations that the animal studies might miss? Effects below the NOAEL. You hear concerns about that low dose range.
That's a major concern, absolutely, and one of the biggest challenges. Animal studies are usually designed to detect maybe a 10% to 15% increase in a toxic outcome to be statistically significant. This leaves considerable uncertainty about what happens below the NOAEL. Toxicologists often assume a threshold dose below which there's no toxicity, implying that cellular defenses can handle it up to a certain point. But the exact shape of the dose-response curve below the NOAEL is often unknown. And for some toxicants, particularly those that directly damage DNA, there might not be a threshold at all. Any exposure, theoretically, could carry some risk. Ideally, mechanistic studies should predict the most likely dose-response curve shape for each chemical.
In mechanistic studies, that's like really digging into the fundamental how of it all. How does a chemical actually cause harm at the molecular level?
Precisely. Toxicologists perform these studies to understand how a chemical causes toxicity, allowing for more accurate predictions and interventions. There are several tools: computer modeling, like QSR, quantitative structure-activity relationships. These use a compound's 3D structure and properties to predict toxicity or binding to specific targets. A rapid screening method.
Using computers to predict harm.
In a way, yes. Then there are cell-based approaches, using bacteria or human cells to see if a compound damages DNA or kills cells. The Ames test is a classic example. It uses mutated bacteria that can't grow without a specific amino acid. If a chemical is mutagenic, it fixes the mutation and the bacteria grow.
Clever.
Often done with and without metabolic activation like liver enzymes to see if the body makes it more toxic. Then you have the omic approaches. High-throughput tech like RNA sequencing, transcriptomics, proteomics, metabolomics. They look at all the changes in RNA, proteins, or metabolites in response to a toxicant. Gives a systems-level view.
Seeing the whole picture.
Exactly. And finally, knockout models. Using cells or animals where a specific gene is disabled. If the toxicity disappears in the knockout model, it strongly suggests that gene is involved in the mechanism. Provides strong causal evidence. Understanding these precise molecular mechanisms, you see, helps us move beyond just observing effects to truly predicting and preventing harm. If you know how something is toxic, you can design better ways to block it or mitigate its effects. It's really foundational knowledge for mastering this field. 3. Cancer and prevention. The silent battle within.
Cancer, for so many of us, that's one of the most frightening potential outcomes of chemical exposure. How do scientists classify these agents, these carcinogens, and what does it really mean for a chemical to cause cancer?
That's a crucial question, and the classification is rigorous. The International Agency for Research on Cancer, IARC, part of the WHO, is a leading authority. They classify carcinogens into groups based on comprehensive risk assessments, integrating all available data, human epidemiology, exposure data, animal studies, and detailed mechanistic data. It's a very thorough process.
So what are the groups?
Okay, Group 1 are known human carcinogens. The evidence is considered conclusive. Examples include things like nitrosamines in tobacco, benzocaine from combustion and smoke, some aromatic amines from dyes, and aflatoxin B1, that fungal toxin. Interestingly, this group also includes nongenotoxic agents like ethanol, certain phorbol esters, estrogens, metals like arsenic, irritants like asbestos, and dioxins like TCDD. So the mechanism can vary even if the outcome is cancer.
Group 1 is the highest risk then. What's next?
Group 2A are probably carcinogenic in humans. Strong evidence, maybe not quite as definitive as Group 1. Group 2B are possibly carcinogenic. Weaker evidence, often limited, but suggests a potential link worth investigating. Group 3 means the data are insufficient to classify its carcinogenicity. More research needed. And Group 4 means data indicate they're unlikely to be carcinogens. This is quite rare.
The journey to malignancy.
Now, the process of a normal cell becoming cancerous. It's anything but simple. It's complex, multi-stage, usually unfolds over a long time, and environmental chemicals can act at one or more of these stages. A classic, though simplified model involves tumor initiation, then tumor promotion, and finally tumor progression.
Initiation, promotion, progression.
Right. In this model, an initiator causes permanent genetic damage, usually gene mutations. These mutations give a cell an advantage, maybe it proliferates faster, or it avoids programmed cell death, apoptosis.
So it gets a head start and refuses to die.
Exactly. A promoter, on the other hand, doesn't directly damage genes. Instead, it alters signaling pathways or the cell's environment to boost the survival and proliferation of these already initiated precancerous cells.
Yeah.
Think of the initiator planting an abnormal seed and the promoter fertilizing it.
Helping it grow.
Precisely. Ultimately, over time, more chromosome instability and additional mutations accumulate, leading to invasiveness and metastasis, the hallmarks of full-blown cancer spreading beyond the original site.
So it's not just a single hit, but this whole cascade. How do chemicals fit into this precise multi-step process?
Chemical carcinogens typically cause cancer through two main mechanisms, genotoxic and non-genotoxic.
Let genotoxic first, damaging genes.
Yes. Genotoxic carcinogens initiate tumors by directly damaging DNA. They often get metabolized in a target tissue into a reactive intermediate. This intermediate can then directly stick to DNA, forming a DNA adduct, which messes up the DNA structure. Or it can indirectly damage DNA by forming reactive oxygen species, ROS, free radicals, which oxidize DNA or create lipid peroxidation products that then react with DNA. If this damage isn't repaired before the cell divides, a permanent mutation can occur. If that mutation hits a key tumor suppressor gene, the brakes, or a proto-oncogene, the accelerator, it gives the cell a growth or survival advantage. Mutations in DNA repair genes also make future mutations more likely. With the right conditions, these mutated cells can proliferate and become cancer. A prime example is benzoypyrene, found in tobacco smoke, exhaust, charbroiled food. It gets oxidized by CYP enzymes to a more carcinogenic metabolite, 7-molly-8 dihydrodiol. This can then be oxidized again to form a diol epoxide, which readily reacts with DNA. Or it can form a catechol, which generates ROS, so it damages DNA directly and indirectly.
Okay, and the other type, non-genotoxic.
Right. Non-genotoxic carcinogens increase cancer risk without directly damaging DNA. Most act as tumor promoters. They don't initiate by mutation, but they create an environment where initiated cells thrive. Many bind to receptors that stimulate proliferation or other promoting effects like tissue invasion or angiogenesis, growing new blood vessels to feed the tumor.
Like fertilizer for a bad seed.
Exactly. For instance, phorbol esters mimic a natural signaling molecule activating protein kinase C, PKC, which stimulates pathways leading to proliferation and invasion. Normally, this might trigger cell death, but initiated cells with defective apoptosis survive and grow. Estrogenic carcinogens activate the estrogen receptor alpha, ERA, stimulating growth in estrogen-responsive tissues like breast or uterus. Chronic inflammation is another big one. Inflammatory signals stimulate PKC. Irritants like asbestos cause chronic lung inflammation, promoting cancer. Even chronic liver damage from toxins or hepatitis stimulates compensatory proliferation, which increases mutation risk and selects for faster-growing cells.
So some chemicals could do both, initiate and promote, a double whammy.
Precisely. Many compounds act as complete carcinogens because repeated exposure can cause both initiation and promotion. Benzoaperine and UV radiation are classic examples. They damage DNA and promote the growth of damaged cells.
And what about things that don't hit DNA directly but still set the stage, like making DNA more vulnerable or changing gene control?
You're honing in on a critical, more subtle aspect. Initiation can also happen through non-genotoxic mechanisms. Some heavy metals interfere with proteins needed for DNA synthesis and repair, increasing replication errors without directly damaging DNA. Non-genotoxic carcinogens can also cause heritable changes in gene expression without changing the DNA sequence itself through epigenetic effects. They can alter DNA methylation patterns, silencing tumor suppressor genes or activating proto-oncogenes. These changes can be passed to daughter cells.
So changing how genes are read, not written.
Exactly. Furthermore, some chemicals act as co-carcinogens. They don't cause cancer themselves, but they enhance the carcinogenicity of other chemicals. They might increase absorption, alter metabolism to create more toxic forms, or interfere with DNA repair, making another carcinogen much more potent. Ethanol is a key example. It acts as a solvent, increasing absorption of tobacco carcinogens in the mouth and throat, dramatically raising head and neck cancer risk for drinkers who smoke. Ethanol also depletes glutathione, GSH, limiting detoxification of reactive metabolites and ROS. It raises an important point. In the real world, it's rarely just one chemical. Often it's the combination and interaction that drives risk.
That's a powerful and alarming interaction. But if chemicals can cause cancer, can other agents prevent it? This is where chemoprevention comes in, right? A more proactive approach.
Proactive protection against cancer.
This is indeed where the exciting field of chemoprevention comes in. These are drugs or natural agents that interfere with the carcinogenic process to prevent cancer before it's ever diagnosed. The concept emerged back in the 1960s with observations that certain dietary components could prevent cancer in rodents. Many modern strategies are based on epidemiological studies showing protective effects of plant-based foods and drinks against various cancers. The hope is to isolate these active compounds and develop preventive drugs.
So what does this all mean for us? We're looking for a proactive defense, not just a treatment. And how successful has this been?
Exactly. Chemoprevention is still an emerging field with many compounds in clinical trials. But it's challenging. Many tested chemicals haven't shown a benefit, and some, unexpectedly, have even increased cancer risk. Currently, there are no FDA-approved drugs specifically for preventing environmental carcinogenesis. However, there are approved drugs for preventing cancer due to endogenous estrogen, like tamoxifen and raloxifene for breast cancer.
Right, those are well known.
And highly successful vaccines prevent cancer caused by viruses. The hepatitis B vaccine prevents liver cancer, and HPV vaccines prevent cervical and other cancers. These successes provide important proof of principle that chemoprevention is possible when targeting specific mechanisms.
How do these chemopreventative agents actually work at a molecular level? What are their strategies?
They can interfere with both initiation and promotion through several mechanisms. Anti-initiation. One way is preventing carcinogen activation. Isothiocyanates from cruciferous vegetables like broccoli inhibit CYPs that activate many carcinogens. They also upregulate genes controlled by the antioxidant response element, ARE, including those for GSH synthesis and quinone reductase, boosting detoxification.
So stopping it before it starts or cleaning it up quickly.
Exactly. Antioxidant effects. Many carcinogens work by generating ROS, so antioxidants offer protection. Flavonoids and polyphenols from plants, green tea, berries, are potent antioxidants that also inhibit CYPs and induce ARE genes. Binding or reacting with carcinogens. Some compounds, like chlorophyll, can bind tightly to carcinogens like aflatoxin in the GI tract, preventing absorption.
Intercepting them.
Right. Interfering with promotion, inflammation is a key target. COX-2 inhibitors like celecoxib reduced colorectal cancer risk, but cardiovascular side effects halted trials. However, long-term aspirin use reduces colorectal adenomas and cancer mortality. Shows the potential, but also the risks. Some natural compounds, like atocoferol, vitamin E, were hoped to reduce inflammation, but actually increased prostate cancer risk in one trial. It highlights the complexity.
Unexpected outcomes.
Definitely. Disrupting nuclear receptor signaling. Retinoids showed early promise for head and neck cancers, but later increased lung cancer risk. In contrast, selective estrogen receptor modulators, SERMs, like tamoxifen and raloxifene, successfully reduced breast cancer incidence in high-risk women and are FDA approved for chemoprevention. This success shows that mechanism-based drug development can work for prevention.
This is a fascinating intersection of diet, environment, and pharma. Let's do a deep dive into a specific example that brings many concepts together. Aflatoxin B1.
Aflatoxin B1, a foodborne carcinogen, and its prevention.
Aflatoxin B1 is a potent hepatocarcinogen and causes liver cancer. It's produced by Aspergillus fungi, common in hot, wet climates, contaminating foods like corn, peanuts, cottonseed, tree nuts. It's a serious problem in parts of Latin America, Africa, and Southeast Asia, leading to high rates of human hepatocellular carcinoma, HCC. While exposure in the U.S. is rare due to regulations, it's a critical global health concern.
So how does this toxin get in and cause damage? It sounds like just a mold toxin.
Unfortunately, it's quite efficient. Aflatoxin B1 is readily absorbed from the GI tract and goes straight to the liver for first-pass metabolism. Here's the problem. Specific CYP enzymes, CYP1A2 and CYP3A4, metabolized it either to an 8-phthalate-9-epoxide, the reactive, DNA-damaging carcinogenic intermediate, or to less harmful detoxification products like aflatoxin M1 and Q1. That 8-phthalate-9-epoxide is highly reactive. While some gets detoxified by hydrolysis or conjugation with glutathione, GSH, a significant portion quickly reacts with DNA and proteins, forming damaging adducts. The detox product, aflatoxin M1, enters circulation and is excreted in urine and milk.
What are the health consequences then? What does it actually do?
It primarily targets the liver, causing cancer, but also affects the GI tract and blood system. High-dose exposures, though rare, cause acute liver necrosis, jaundice, often death, but chronic exposure is the widespread global issue, leading to liver cirrhosis and immunosuppression. IRC classifies it as a Group 1 known human carcinogen based on strong human, animal, and mechanistic data. And here's where it gets really interesting and highlights a critical public health synergy. Aflatoxin exposure and the hepatitis B virus work together powerfully to cause liver cancer. It's multiplicative. Aflatoxin alone increases risk 3.4-fold, hepatitis B alone 7.3-fold. But individuals exposed to both have a staggering 59-fold increased risk compared to unexposed people.
59 times. Wow, how does that happen?
Several mechanisms. The hepatitis B virus X gene product, HBX, inhibits P53, suppressing DNA repair of aflatoxin adducts. Hepatitis B also alters aflatoxin metabolism, upregulating CYP3A4, more toxic epoxide, and decreasing GSHS transferase activity, less detoxification. Plus, the chronic liver inflammation and compensatory cell proliferation caused by hepatitis B creates an environment where aflatoxin-induced mutations are more likely to occur and initiated cells are more likely to grow.
That's a powerful example of how factors compound. Can we prevent this double threat?
Absolutely. The link between its metabolism and carcinogenicity makes it a prime target for chemoprevention that modifies metabolism. The goal is to reduce that harmful 8-methionine epoxide. Several ways. Ultipraz, an old drug, inhibits CYPs and induces our RE genes for GSH synthesis. It increased excretion of the detoxified aflatoxin conjugate in human studies. Green tea polyphenols. Daily doses equivalent to 1 to 2 liters of green tea showed a decline in aflatoxin albumin adducts and increased excretion of the detox conjugate. Protective effect. Another strategy uses interceptor molecules like chlorophyllin, water-soluble chlorophyll salts. It binds a phthatoxin in the GI tract, preventing absorption. A phase 2 trial showed 100 milligram with meals reduced aflatoxin DNA adducts in urine. And finally, because of that strong synergy, the hepatitis B vaccine is incredibly effective indirectly. Preventing Hep B dramatically reduces sensitivity to aflatoxin cancer. Plus, primary prevention hand sorting crops, better food storage to limit mold growth, these are cost-effective public health strategies. 4. The polluted planet. Air, water, and ubiquitous metals.
Okay, shifting gears now to the broader environment. Agencies like the EPA try to keep our water and air clean. What's involved there? How do they set standards for the huge number of potential contaminants out there?
The U.S. Environmental Protection Agency, the EPA, plays a crucial role, enforcing major laws like the Clean Water Act, Safe Drinking Water Act, and Clean Air Act. Under these laws, they use a complex process combining scientific risk assessment with economic analyses and other factors to develop regulations on pollutant release or presence. The goal is limiting exposure below levels considered safe for health and the environment. It's a balancing act.
Okay, like for drinking water.
For drinking water, toxicity screening leads to maximum contaminant levels, MCLs. A primary MCL is an enforceable limit based on the toxic dose-response curve and whether we can reliably measure it. A secondary MCL isn't enforced but addresses aesthetics, taste, color, odor. There's current concern about pharmaceutical waste and water, but no MCLs have been set for those yet due to the complexity.
And for air?
For air, the EPA monitors 6 priority pollutants: carbon monoxide, CO, lead, PB, nitrogen oxides, NOx, ozone, O3, particulate matter, PM, and sulfur dioxide, SO2. They set national ambient air quality standards, leading to daily air quality reports and warnings like ozone action days.
I often hear about PM 2.5 specifically. Why is that particular size such a concern?
Ah, yes. Fine particles smaller than 2.5 micrometers in diameter, PM 2.5, are particularly worrying because they're so tiny. They penetrate deep into the lungs, bypassing our natural defenses, reaching the alveoli where gas exchange happens.
So they get right in.
Exactly. And they can even cross into the bloodstream. Once there, they cause or worsen respiratory illnesses like asthma and COPD and cardiovascular problems like heart attacks and strokes. Plus, other toxic chemicals often stick to these tiny particles, hitching a ride deep into the body.
Beyond air and water, metals are a big deal, right? Arsenic, lead, mercury. They seem so persistent and have such diverse effects.
Absolutely. Metals are a crucial class of environmental toxicants from natural and human-made sources. The CDC lists arsenic, mercury, and lead as the top 3 environmental pollutants of concern based on toxicity and human exposure levels. While high-level toxicity has been known for centuries, the effects of low-dose chronic exposure are only recently fully appreciated. Many are also carcinogens, and even essential metals like copper and iron can be toxic at high concentrations, often targeting the liver via ROS generation.
A deeper look.
The lingering threat.
Lead poisoning. Feels like history, like Roman aqueducts or old paint. But it's clearly still very relevant today. Why is that, and what makes it so insidious?
It absolutely is relevant, a testament to its persistence. Despite huge reductions over the past 4 decades, lead exposure remains a major concern, especially for children. Lead paint was banned in U.S. homes in 1978. Leaded gasoline phased out by 1996. That drastically cut air lead. But lead doesn't degrade. It persists in dust, soil, and paint in older homes.
So it's still there?
Yes. Young children are vulnerable hand-to-mouth behavior, ingesting paint chips or dust. Renovations stir it up. Historical fallout remains in urban soils. And lead pipes can leach into drinking water. We've seen that tragically in places like Flint. Other sources exist too: firing ranges, folk medicines. Blood lead levels, BLLs, in kids are way down from maybe 15 EGDL in the 70s to around 1.3 now. But critically, the CDC now says there is no safe level of lead exposure for children. Any exposure is concerning.
No safe level at all?
None. The focus is now on primary prevention for all kids and identifying those in the top 2.5% of exposure, currently in low 5 DDL, for intervention and screening. Chelation therapy is still used for very high levels, above 45 big DL.
So if there's no safe level, what happens when lead gets in? How does it cause harm?
Lead is usually divalent, Pb2 plus 10. Its toxicity comes mainly from molecular mimicry. It looks and acts like other essential divalent metals, especially zinc and calcium. Because it's similar in size and charge, it can substitute for them in proteins, disrupting their structure and function, interfering with crucial biological processes.
Okay, and how does it get absorbed and move around? ADME.
Exposure is via ingestion or inhalation. GI absorption varies. Kids absorb about 40%, adults less than 20%. Fasting, or calcium iron deficiency, increases absorption. It likely uses the same transporters. Big issue for malnourished kids.
Ah, so deficiency makes it worse.
Yes. Inhaled lead absorption is much higher, around 90%. Organolead, like tetraethyl lead, could be absorbed through skin, but inorganic lead isn't much. In blood, 99% binds to hemoglobin. It distributes first to soft tissues, kidney, liver, brain, then slowly redistributes and deposits mainly in bone, teeth, hair. About 95% of adult lead burden is in bone.
95%?
Yes, it can show up as lead lines on kids' x-rays. Bone lead slowly leaches back into blood, especially during calcium stress like pregnancy, potentially exposing the fetus. Small amounts get into the brain, especially gray matter and basal ganglia, and it readily crosses the placenta.
How is it eliminated?
Primarily in urine, proportional to the free lead in plasma, only 1% of total BLL. Smaller amounts in milk, sweat, hair, nails. Serum half-life is 1-2 months. But in bone, the half-life is estimated at 20-30 years. It persists for decades.
That persistence, especially in bone, affects decades later. What are these pervasive health effects, even at low levels?
Right. While high-dose poisoning is obvious, the toxicities of chronic low-dose lead, BLL-20, big DL, are more insidious but well understood now. Lead affects multiple systems, but the nervous, hematological, blood, cardiovascular, and renal kidney systems are most sensitive.
Nervous system first, especially kids.
Yes, the greatest concern is cognitive delays and behavioral changes in children. The developing nervous system is exceptionally sensitive, with effects seen even at the lowest measurable BLLs. Lead inhibits calcium transporters and channels, alters calcium-responsive proteins like PKC and calmodulin. This disrupts neuron activation, neurotransmitter release, affecting dopaminergic, cholinergic, glutaminergic systems vital for learning, memory, mood. At high levels, it disrupts the blood-brain barrier.
How does it affect brain development itself?
It interferes with synapse pruning, neuronal migration, neuron-glial interactions. This can result in decreased IQ, poor exam performance, language motor deficits, behavioral issues like distractibility, impulsivity, aggression, short attention span. No evidence for a threshold. Very high BLLs, 70 big DL, risk encephalopathy, lethargy, vomiting, irritability progressing to ataxia, delirium, coma, death. High mortality, long-term neuro problems for survivors. Adults are less sensitive but can get encephalopathy. Chronic exposure historically caused lead palsy, wrist-foot drop from motor neuron degeneration. Studies link lead exposure in older adults to faster cognitive decline.
What about heart and kidneys?
Elevated blood pressure is a lasting effect of low-level lead, BLL-10 mg/dL, even from childhood exposure. Lead increases risk of death from cardiovascular and cerebrovascular disease. In the kidney, low levels, 10 mg/dL, depress glomerular filtration. Higher levels cause proteinuria, impaired transport. Very high 50 cause permanent damage like nephropathy, glomerulosclerosis. Kidney damage and high blood pressure are closely linked. Cardiovascular effects might involve ROS production reacting with nitric oxide, NO, preventing vasodilation. Kidney effects might involve mitochondrial damage.
And other effects.
Good name, sister.
Lead causes immunosuppression and inflammation, altering T-cell and macrophage signaling even at low BLLs. It inhibits enzymes in heme biosynthesis seen even below 10 mg/dL. Chronic high doses cause hypochromic microcytic anemia, similar to iron deficiency. Acute high dose causes lead colic, severe gut pain.
Is it linked to cancer? IRC classifies lead as group 2A, probably carcinogenic to humans. Studies link it to lung, brain, kidney, stomach cancers. Rodents get kidney tumors, gliomas. Lead isn't directly mutagenic, but increases chromosomal damage, clastogenic. Carcinogenesis might involve inhibiting zinc finger proteins needed for DNA repair and synthesis, a non-genotoxic mechanism.
So if someone's exposed, what's the treatment? Can we reverse the damage?
Most important, remove the source of exposure, always. Supportive care for symptoms. Chelation therapy is warranted for very high BLLs, 45 VIG-DL kids, 60 adults, or acute symptoms. Oral chelation for BLLs 45-70 in kids. Above 70 in a child is a medical emergency needing hospitalization and IV chelation. But here's the crucial caveat. While chelation lowers BLL and helps acute symptoms, it does not reliably reduce the chronic effects beyond just stopping exposure. In rats, some chelators might even increase lead going to the brain.
So it's not a cure-all for the long-term damage.
Exactly. Its use needs careful consideration.
The liquid metal.
Mad as a hatter. That phrase instantly comes to mind with mercury. What's its story, and how is it still a modern concern?
That phrase indeed came from Hatter's chronic exposure to metallic mercury vapor-making felt hats. Mercury is unique liquid at room temp, used for millennia because it amalgamates with other metals. Hippocrates noted its toxicity. Its use for syphilis inspired Paracelsus's famous line, "The dose makes the poison."
How are people exposed today?
Inorganic and metallic mercury occur naturally, released by volcanoes, off-gassing. But human activities like fossil fuel burning, coal, and gold mining release huge amounts. The EPA set standards for coal plants in 2011. Airborne metallic mercury gets oxidized, deposits in water via rain. Microorganisms there transform it into methyl mercury, MeHWL. This concentrates in tissues and bioaccumulates up the food chain. That's why top predators like swordfish and sharks have high levels.
So fish is a major source.
Yes, organic mercury MEG plus exposure is primarily from fish consumption. The general population's main source of elemental mercury, HG0 vapor, is often from dental amalgam fillings. Workers get exposed to HGO vapor and inorganic mercury in industries like chlor-alkali production, chemical processes using mercury catalysts, making devices like batteries, bulbs, thermometers, dentists too from amalgam.
What about that thimerosal controversy in vaccines?
Yes. Thimerosal is an antimicrobial preservative containing ethyl mercury, similar to MEG plus process. Concerns about autism links were based on discredited reports. It was removed from most U.S. childhood vaccines in 2001, largely due to public pressure, but huge studies found no link to negative outcomes. It's still safely used in multi-dose flu vaccines.
Okay, so what are the key forms and how do they act?
3 key forms: metallic, elemental mercury, Hg0, the liquid metal, volatile vapor; inorganic mercury, mercurous, Hg1+, or mercuric, Hg2+, forms salts. Organic mercury, like methyl mercury, MEdH+, the main environmental concern. Both Hg2+ and MEdH+, readily form strong covalent bonds with sulfur, especially sulfhydryl groups on proteins. This disrupts protein function. Microtubules are particularly sensitive. There might also be an autoimmune component.
How about ADME for these forms?
Hg0 vapor, readily absorbed via lungs, 70–80%, minimal GI absorption, distributes widely, crosses blood-brain barrier and placenta, oxidized in cells to Hg2+ by catalase, some exhaled, can be oxidized to Hg2+ in the brain and retained. Inorganic mercury salts, GI absorption 10–15%. Hg1+ can form Hg0 or Hg2+. Hg2+ mainly excreted in urine and feces. Half-life 1 to 2 months. All forms excreted in sweat, milk, hair, nails. Mej plus art. Complexes with cysteine, mimics methionine, transported across membranes efficiently. Almost completely absorbed from GI tract, 90%. Readily crosses blood-brain barrier and placenta. Distributes fairly evenly, highest in kidneys. Can be slowly demethylated to Hg2+. Excreted via urine feces. Feces dominates. Half-life about 2 months. Molecular mimicry is key to its toxicity.
And the health effects, they vary by form.
Significantly, metallic mercury, Hg0. Acute high vapor inhalation is toxic to lungs. Cough, pneumonitis. Other symptoms: weakness, chills, metallic taste, GI upset. Chronic exposure hits the nervous system. Tremors, hands especially. Emotional applicability, irritability, shyness, Mad Hatter. Insomnia, memory loss, cognitive deficits. Worsens and becomes irreversible. Also kidney damage, tachycardia, salivation, gingivitis, inorganic salts. Ingestion irritates GI tract intensely. Acute exposure, for example, suicide attempts, causes renal tubular necrosis, acute renal failure. Chronic exposures target the kidney, causing glomerular injury. Organic mercury, MANH+. CNS is the primary target. High doses cause visual disturbance, ataxia, paresthesia, fatigue, hearing loss, cognitive deficits, paralysis, death. The developing nervous system is especially sensitive. In utero exposure can cause severe intellectual disability, neuromuscular deficits, even if the mother is asymptomatic.
That's a stark difference depending on when exposure happens. Very concerning.
It is. And assessing low-dose MEG+ from fish is tough due to the beneficial effects of omega-3s in fish oils. Leads to conflicting study results. It raises that important question: how do we balance benefits and risks?
What about treatment?
Terminating Hg0 exposure is critical. May need respiratory support. Emesis might be used for acute inorganic salt ingestion soon after, if safe. Maintaining fluids electrolytes is vital. Chelation therapy helps patients with acute inorganic or metallic mercury exposure. Important. Limited options for MEG+ OP. Chelation does not provide clinical benefits. Non-absorbable thiol resins might help prevent GI absorption, but don't treat absorbed mercury. Regarding fish intake, restricting it for women of reproductive age children is controversial due to omega-3s. EPA recommends limiting to 12 ounces a week. Many feel this is too conservative, but the advice to eat lower mercury fish, canned light tuna, salmon, pollock, and avoid top predators, swordfish, shark, is widely accepted.
The ancient poison, modern concern.
Arsenic. Such a dramatic history. Poisons, chemotherapy. How does this metalloid fit into modern environmental concerns?
Indeed. Arsenic, a metalloid common in rock soil, used as therapeutic and poison for over 2,400 years. Robert Ehrlich coined magic bullet in chemotherapy for his arsenic compound for syphilis. While mostly phased out of drugs, arsenic trioxide is still effective chemo for acute promyelocytic leukemia, shows its dangerous duality.
Where does exposure come from today?
Primary source globally is drinking water, leaching naturally from rock soil into wells. U.S. average is low, 2 IGL, but private wells can exceed the EPA MCL of 10 IGL significantly. Some global regions, Taiwan, Bangladesh, India, have vastly higher levels, causing widespread poisoning. Human activities like arsenic pesticides, mining, coal burning also release it. Food, especially seafood, can be contaminated, but usually less toxic organic forms. Average intake 10 mg/day, mainly food water. Before 2003, pressure-treated wood used lots of arsenic, safe unless burned. Occupational exposure from herbicide-insecticide production, high-tech industries, computer chips.
What are the chemical forms and modes of action?
Arsenic exists as elemental, trivalent, arsenides As3+, pentavalent, arsenates As5+. Arsine, ASH3, is a highly toxic gas, Organic forms too, toxicity generally. Organic AS5 plus AS3 plus ASH3. Trivalent is more toxic than pentavalent. Like mercury, trivalent arsenic AS3 plus binds sulfhydryl groups. Pyruvate dehydrogenase complex, energy metabolism, is sensitive. Arsenic inhibits it via lipoic acid cofactor. Pentavalent arsenate, AS5 plus, inhibits electron transport by substituting for phosphate in ATP formation, creating unstable esters. Disrupts energy production.
And ADME, absorption, distribution.
Yeah. Absorption depends on water solubility. Water-soluble forms readily absorbed via inhalation ingestion, 90% from drinking water. Low doses distribute evenly, high concentrations in nail share, high sulfhydryl.
Yeah.
Acute high doses deposit in liver kidney, crosses placenta and blood-brain barrier. Arsenic undergoes biotransformation. As3 plus can be oxidized to As5 plus Sin. Humans methylate, it produces MMA and DMA. While pentavalent methylated forms are less toxic, trivalent methylated arsenicals, like MMAII, are more toxic than inorganic AS3 plus due to higher sulfhydryl affinity. So methylation can be a bioactivation pathway.
That's counterintuitive. How is it eliminated?
Primarily in urine, mixture of inorganic monodimethylated forms, also feces, sweat, hair, nails, skin. Excreted relatively quickly, half-life 1-3 days.
Health effects. Sounds like it hits everything.
Pretty much, except arsine gas, which is distinct. Inorganic forms have similar broad toxic effects. Humans are most sensitive. Acute large doses, 70 to 180 milligrams, often fatal. Heart, GI effects, multi-organ failure. Cardiovascular. Acute to chronic exposure causes arrhythmias, ischemic heart disease. Chronic exposure can cause peripheral vascular disease, like Blackfoot disease in Taiwan. Cyanosis, gangrene. Arsenic increases capillary permeability, edema acutely, vascular disease chronically. DNA methylation might play a role. Skin, often diagnostic. Hyperkeratinization, cornswords, palm soles. Hypopigmentation, raindrop pattern. Can appear after month years with 100 EGL in water. Kids more prone. GI tract. Acute subacute high dose causes cramping to hemorrhaging, death, capillary permeability, fluid loss, not seen with chronic low-level exposure. Nervous system. Common effect is peripheral neuropathy, sensory motor loss, weakness. Appears days after exposure, often incomplete recovery. May cause intellectual deficits in children. Acute high dose can rarely cause encephalopathy, headache, confusion, seizures, coma. Other non-cancer. Anemia, leukopenia, cytotoxicity, suppresses blood formation. Liver effects, fatty infiltration, necrosis, cirrhosis. Kidney damage. Lung irritation, inhaled. Bronchitis, ingested. Increased diabetes risk. Carcinogenesis. High arsenic and water linked to skin, bladder, lung cancer. Associations with liver, kidney, prostate tumors. Inhalation causes lung cancer. IRRC classifies arsenic as group 1 carcinogenic to humans.
So what does this mean for the most vulnerable, like children and developing fetuses?
Critical concern. Developing fetus young children may have increased risk of arsenic-mediated cancer later, including lung cancer. Rodent studies show increased risk from in utero exposure, suggesting the second trimester is a critical window. Arsenic doesn't directly damage DNA, not mutagenic. Thought to work via changes in gene expression, DNA methylation, epigenetics, inhibiting DNA repair, oxidative stress, altered signal transduction. Can be a tumor promoter or co-carcinogen. Potentiates lung cancer from tobacco. Smokers in high arsenic regions have 5x risk over smokers in low arsenic regions. May involve inhibiting nucleotide excision repair. Also has endocrine disrupting activities, steroid hormone receptors. Arsine gas, rare but often fatal, formed by arsenic reduction industrially. Induces rapid hemolysis, red blood cell destruction. Symptoms, headache, vomiting, abdominal pain, dark urine, kidney failure. 25% fatal.
Treatment options.
Stabilize patient, prevent further absorption. Fluid monitoring crucial, risk of shock. Chelation therapy helps short-term acute exposure, little benefit for chronic. Exchange transfusion often needed for arsine gas.
A corrosion-resistant toxicant.
Cadmium seems to be in batteries, paints, plastics. What makes it a concern and what are its key toxic effects?
Cadmium, discovered 1817, widely used since mid-20th century for corrosion resistance, electrochemical properties. Used in electroplating, galvanizing, plastics, pigments, nickel-cadmium batteries.
Where does exposure come from?
General population. Primary source is food, 50-day intake. Also tobacco smoke, 1 to 2 big cigarette. Workers in smelters, metal processing face high exposure, especially inhalation.
Chemistry and mode of action.
Cadmium is a divalent cation, CD2+, doesn't undergo redox. Toxicity mechanism not fully clear. Like lead, can replace zinc in zinc finger proteins, disrupting function. Induces ROS formation, lipid peroxidation, GSH depletion, upregulates inflammatory cytokines, disrupts NO effects.
ADME for cadmium.
Poorly absorbed from GI tract, 1.55%. Better via inhalation, 10%. Distributes first to liver, then kidney, together account for 50% of dose. Doesn't cross blood-brain barrier or placenta significantly. Primarily excreted very slowly in urine. Remarkably long half-life, 10-30 years. Bioaccumulates significantly.
8 to 30 years, wow.
Yeah.
Health effect.
Toxicity. Acute cadmium causes local irritation. Inhaled. Severe respiratory irritation, pneumonitis, potentially fatal pulmonary edema. Ingested. Nausea, vomiting, salivation, diarrhea, abdominal cramps, often bloody. Chronic toxicity depends on route. Lung is major target for inhaled, kidney for both inhalation and ingestion. Cadmium transported to kidney, bound to metallothionein, released there. Renal toxicity causes excretion of low molecular weight proteins, glomerular injury, decreased filtration. Chronic occupational exposure linked to renal failure, death. No evidence for a threshold for kidney effects. Even dietary levels can cause renal toxicity. Workers with long-term inhalation show decreased lung function, bronchitis, fibrosis, emphysema, increased COPD mortality. With vitamin D deficiency, cadmium increases fracture osteoporosis risk, interferes with calcium phosphate via renal toxicity. Led to Itai disease in Japan, carcinogenicity. Chronic occupational inhalation increases lung cancer risk, IRC group 1. Mechanism unclear, but causes chromosomal aberrations, increases mutations, impairs DNA repair, substitutes for zinc and repair proteins, inhibits nucleotide excision repair, alters cell signaling. Acts as nongenotoxic carcinogen.
Treatment for cadmium.
Symptomatic, respiratory support for inhalation, kidney transplant for failure. Importantly, no clinical benefit from chelation therapy for cadmium, may even cause harm.
The metal with multiple faces.
Chromium, common in stainless steel. May be thought to be essential. How does it become a toxic problem?
Chromium is industrially vital, especially for stainless steel, 11% Cr. Exists in multiple valence states, but trivalent, CrEI, and hexavalent, CrVi, are biologically important. Chromium is stable, common in nature, once thought essential for glucose metabolism, now largely refuted. CrVi is highly corrosive, generally responsible for toxic effects. Valence state is key.
Exposure routes.
General population, mainly food, also drinking water, air. Workers exposed during chromate production, stainless steel work, plating, tanning. Often a mix of Craig or Affy.
Chemistry and mode of action. How does CrVi cause harm?
CrER is stable, low toxicity. CrVi is corrosive, readily reduced inside cells. Differing toxicity mainly due to absorption distribution differences. Hexavalent chromate, CrVi, resembles sulfate phosphate, enters cells via anion transporters. Inside, it's reduced, ultimately forming CrIRV. This intracellular CrI is thought to cause most toxicity by forming covalent bonds with DNA, proteins. Hexavalent chromium also induces oxidative stress, hypersensitivity reactions.
ADME for chromium.
Inhaled absorption depends on solubility, valence, particle size. Soluble CrVi absorbed more than insoluble Cri, 50–85% of small CrVi particles absorbed. Ingested absorption 10%. CoVi crosses membranes via transport, CrIR via diffusion. CrVi distributes widely, crosses placenta, highest in liver, kidney, bone. Excreted mainly via urine. Half-life of ingested CrVi, 40 hours CrIR time. Cleared relatively quickly.
Health.
Toxicity. Acute high doses cause death, kidney damage. Chronic low dose causes toxicity at contact site. Inhaled, lung respiratory irritation, decreased function, pneumonia. Ingested inclcurd, inicles, irritation, ulcers, diarrhea, pain, vomiting. Dermal. CrVi is irritant, causes ulcers, burns. Low-dose exposure can sensitize. Allergic dermatitis, skin, asthma inhalation. Carcinogenesis. CrVi compounds are group 1 known human carcinogens, IARC. Inhaled CrVi increases lung nasal cancer risk mortality. CrVi in drinking water increases stomach cancer risk. Insufficient evidence for metallic trivalent Cr group 3. Multiple mechanisms. Reduction of CrVi to Cri-I oxidizes cellular molecules, ascorbate, GSH, DNA, etc. Oxidative stress. Cri-I forms covalent DNA adducts, mainly phosphate backbone. These aren't highly mutagenic, but trigger high nucleotide excision repair activity. This might paradoxically cause cancer by preventing repair of other lesions or causing breaks from incomplete repair. Chromium also forms DNA-protein cross-links. Chronic inflammation also promotes tumor. No standard protocols for acute poisoning. Rodent studies suggest reductants, ascorbate, GSH, NAC, before absorption might limit bioavailability by reducing CrVi to Cri-I. These compounds, plus EDTA, also increase urinary excretion after exposure, if given early. Exchange transfusion may help severe cases. 5. Chemical detoxification. The role of chelators.
We've heard chelation therapy mentioned for several metals. So what exactly is a chelator? How do they work? And are they the magic bullet for metal poisoning?
A chelator is a compound that forms stable complexes with metals, typically forming 5 or 6-membered rings. Think of it like a claw, cheli in Greek, grabbing onto the metal ion. The goal is for these complexes to prevent or reverse the metal binding to biological targets like enzymes, and to help excrete the metal, usually via urine. An ideal chelator should be water-soluble, resistant to breakdown, able to reach metal storage sites, form stable non-toxic complexes, and be readily excreted. Ideally, it should have low affinity for essential metals like calcium and zinc.
So are they a magic bullet? Quick fix.
For acute, high-dose exposures to most metals, yes, chelation does reduce toxicity and can be life-saving. However, and this is critical following chronic exposure, chelation often shows no clinical benefits beyond just stopping the exposure. Sometimes it can even do more harm. Some chelators might move metals to more sensitive organs like the brain. So chelation is generally only recommended for acute poisonings or very high blood levels, not as a blanket cure for long-term damage.
At a closer look, ethylene-amidine tetracatic acid, EDTA.
EDTA sounds common. What are its uses and key considerations?
EDTA, particularly Can2-EDD, calcium disodium EDTA, is effective for divalent trivalent metals. The calcium salt is preferred so it doesn't deplete body calcium. It swaps its calcium for metals with higher affinity. Highly effective for acute lead poisoning, often with dimercaprol. However, not effective clinically for mercury or arsenic in vivo.
How does it work?
Chelates metals in extracellular fluid, mobilizing them for urinary excretion. It's charged, so it doesn't enter cells well. Mobilizes some essential metals, zinc, manganese, iron, so zinc supplements might be needed. Used for acute lead intoxication, not beneficial for chronic lead poisoning beyond abatement.
Administration.
Editate calcium disodium. IM injection is absorbed but painful, often mixed with anesthetic. For use requires slow drip in dextrosaline to avoid thrombophlebitis and minimize kidney toxicity.
ADME.
Poorly absorbed orally, 5%. After a 5, short half-life, 20 to 60 minutes, stays in plasma extracellular fluid, excreted rapidly by kidneys. Good renal function essential. Little metabolism. Minimal entry to spinal fluid. Main toxicity is kidney damage. Proximal tubule degeneration. Likely from chelating essential metals like zinc. Reversible usually. Adequate urine output vital. In lead encephalopathy with brain swelling, IV use can fatally increase intracranial pressure. IM preferred then. Rapid the phase of calcium freeform, Ni2-EDA, can cause hypocalcemic tetany. Other side effects: malaise, fatigue, chills, fever, headache, GI issues. Teratogenic in animals, likely zinc depletion. Used in pregnancy only if benefits clearly outweigh risks. Dimercaprol, BAL.
This one sounds like it has a dramatic history. British anti-lewisite.
It does. Developed in WWII as antidote to lewisite and arsenical war gas. Forms stable chelate rings with arsenic, gold, mercury via its sulfhydryl groups.
How does it work and when is it used?
Forms complexes with metals via sulfhydryl groups. Can dissociate. BAL oxidizes in vivo. Bond unstable in acidic urine. Can increase kidney metal delivery, so alkaline urine important. Need continuous dosing for stable 2.1 complex. Avoid high levels due to side effects. Best given soon after exposure. Limits toxicity from arsenic, gold, mercury. Used with Can2-EDTA for lead. Crucially, should not be used for iron, cadmium, selenium complexes are more toxic.
ADME.
Deep IM injection in peanut oil contraindicated if peanut allergy. Peak levels 30-60 minutes. Short half-life, metabolized excreted within 4 H's, urine bile. Contraindicated for chronic exposures, doesn't prevent neurotoxicity, can mobilize lead mercury to the brain in animals due to lipophilicity. Not seen with hydrophilic analogs.
Toxicity sounds significant.
Side effects in 50%. Raises blood pressure, heart rate. Anxiety, nausea, headache, burning sensations, chest pain, conjunctivitis, salivation, sweating, abdominal pain, injection site abscesses. Kids may get fever. Contraindicated in liver failure unless arsenic-induced.
Suxamer.
2.3 dimercaptosacinic acid DMSA.
Suxamer sounds like a newer, better option, maybe oral.
It is a significant improvement. Approved in U.S. for kids with BLL 45 big DL. Orally available, better toxicity profile, selectively chelates. Use off-label for adult lead, arsenic, mercury, though large trials lacking.
How does it work differently?
Chemically similar to BL, 2 sulfhydryl groups, but has 2 carboxylic acids. Modifies absorption, distribution, chelation spectrum. Much better toxicity profile.
ADME, key advantages.
Orally absorbed, biotransformed to mixed disulfide with cysteine. Lowers BLL, attenuates lead toxicity. Chelate eliminated in urine vial, potential enterohepatic circulation. Desirable features, orally bioavailable. Hydrophilic nature means it does not mobilize metals to brain or enter cells readily. Does not significantly chelate essential metals. Zinc, copper, iron. Much better toxicity profile. Animal studies suggest effectiveness for arsenic, cadmium, mercury too, mostly off-label human use.
Toxicity.
Much less toxic than BL. Common effects: mild nausea, vomiting, diarrhea, appetite loss. Rashes can occur, need discontinuation. Transient liver enzyme elevations seen. Sodium 233, dimercaptopropane 1-sulfonate, DMPS, another dimercapto compound. Approved in Germany, available via U.S. compounding pharmacies. Clinically effective for lead, arsenic, especially mercury. Orally available, rapidly excreted kidneys, negatively charged, distribution similar to desoxymer, hydrophilic, less toxic than BL but mobilizes some zinc copper, more than desoxymer. Small trials showed some benefit for chronic arsenic, more studies needed. Penicillamine, trientine.
What about copper toxicity, Wilson disease?
Right. Penicillamine effectively chelates copy, also mercury, zinc, lead urinary excretion. Primary use is Wilson disease, excess copper. Used for other metals too, but considered more toxic, less potent, less selective than others for acute lead mercury arsenic. Not first line. Inexpensive, orally bioavailable, so sometimes use low dose after initial treatment to keep levels down. Well absorbed orally, 40 to 70%, but food and tacit iron reduce absorption. Peak levels 1 to 3 Hrs, relatively stable, metabolized by liver, metabolites in urine and feces. And acetylpenicillamine, more effective for mercury, more resistant to metabolism.
Therapeutic use and toxicity.
Oral, 1.1.5 degree divided doses, empty stomach. Monitor copper excretion for Wilson disease. Toxicity is broad. Skin lesions, rash, lupus, cross-reactivity with penicillin, fatal blood reactions possible, leukopenia, aplastic anemia, renal toxicity, proteinuria to nephrotic syndrome, rare fatal good pasture syndrome, dyspnea, myasthenia gravis reported long-term, requires monitoring. Teratogenic in animals. Benefits outweigh risks for pregnant women with Wilson disease, less serious. GI upset, taste loss, helped by copper supplement. Contraindicated in pregnancy without Wilson. Renal insufficiency, history of blood disorders. Pyridoxine B6, supplement advised. Trientine, acceptable oral alternative for Wilson patients intolerant to penicillamine. Can cause iron deficiency. Need supplements spaced apart from trientine. Deferoxamine, defersirox, deferoprone, iron chelators.
These are specific to iron. Is iron toxicity common?
Can be significant. Acute poisonings, kids ingesting supplements, and chronic overload conditions like thalassemia from frequent transfusions, or hemochromatosis. Deferoxamine, chemistry mode of action, from Streptomyces bacteria, exceptionally high affinity for ferric iron, Fa3+, very low for calcium, removes iron from storage, hemosiderin, ferritin, less from transport and transferrin, does not remove from hemoglobin or cytochromes. ADM use, poorly absorbed orally, needs parenteral. 5-EA, IMSC. Acute toxicity. Iron preferred in less shock than 5E infusion. Chronic overload, thalassemia. Continuous SC infusion best for compliance or slow 5E, not for primary hemochromatosis, phlebotomy referred. Off-label for aluminum chelation, dialysis. Metabolized by plasma enzymes excreted in urine. Toxicity. Allergic reactions, itching, rash, anaphylaxis, dysuria, abdominal discomfort, diarrhea, fever, cramps, and tachycardia. Occasional cataracts, current toxicity, visual auditory changes with long-term high dose. Defersirox, deferoprone. Newer oral chelators FDA approved for chronic iron overload transfusion. Defersiro approves, also for non-transfusion thalassemia. Oral availability improves compliance. Both form uncharged chelate with iron, seem better than deferoxamine at reducing cardiac iron. However, both have FDA black box warnings. Defersirox, renal hepatic failure, GI hemorrhage. Deferoprone, granulocytosis neutropenia, severe low white blood cells. Insufficient data to replace deferoxamine as first line for all chronic overload raises that question. Newer isn't always better. Risks need careful balance. 6. Disrupting internal harmony. Endocrine and immune system targets.
It's clear chemicals can have very specific effects. We've talked metals now, endocrine disruptors. What are they, and why are they so concerning? They mess with hormones, right, which controls so much.
Exactly. Endocrine disrupting chemicals, EDCs, are a broad category. Compounds that interfere with the endocrine system hormone production or signaling. Mechanisms include directly binding regulating hormone receptors like estrogen, androgen, thyroid receptors, their partners, or downstream pathways. Influencing hormone production, secretion, or fate, how they're broken down.
Like throwing a wrench in the body's communication system.
Precisely. Think of hormones as keys, receptors as locks. EDCs can be mimics, wrong key opening lock, blockers, jamming the lock, or just interfering with the process. Beyond reproduction, EDCs impact development, cognitive function, metabolism. Found ubiquitously in plastics, toys, cosmetics, food packaging. Some, like DDT, banned due to no harm, but many putative EDCs remain. FDA identified over 1,800 chemicals with known predicted EDC capacity. Raises the question, How do we track and manage so many potential threats?
A legacy of environmental persistence.
Rachel Carson's Silent Spring, DDT. What was its story and why is it still relevant decades after being banned in many places?
DDT, an organochlorine, was incredibly effective, an insecticide potent, low volatility, high lipid solubility, slow breakdown, hailed as a miracle for malaria agriculture. But those properties, stability, persistence, lipid solubility, became huge environmental concerns. Carson's 1962 book highlighted how DDT persists, bioaccumulates, harms ecosystems like bird eggshells, sparked outrage, led to bans. Sweden 1970, US 1972. While mostly banned, its metabolite, DDE, persists for decades in humans' food chain. DDT is still used in emergencies for mosquito control, so health effects remain relevant.
How are people exposed now?
During use,
Yeah.
40s, 70s, handling inhalation contact. Today, mostly consuming contaminated food where DDT bioaccumulated, fish, fatty foods, leafy root vegetables from contaminated areas too, exposure in utero and via breastfeeding critical, half-life in air 2 days, sunlight breakdown, but in soil 2 to 15 years, DDE still found in most humans worldwide.
Chemistry and mode of action.
Chlorinated ethane, insecticide action, disrupts insect nerve ion channels, Na+, acute human toxicity also via Na+ channels, endocrine effects, OPDDT isomer, 15% is technical grade, is agonist for ERI and EROB, mimics estrogen. PPDDE isomer, main metabolite, is AR antagonist, blocks androgens. BP-Erlig DDT also interacts with FSHR, follicle-stimulating hormone receptor.
EDE.
Exposure, mainly ingestion, DDT, DDE, DDD. High intestinal absorption, increases with fats, distributes widely, accumulates in fat tissue. DDE persists for decades. DDA, oxidized form, main excreted metabolite, urine, feces, breast milk. Combines epidemiology and animal studies. Epidemiology. Serum DDT, DDD consistently correlate with increased risk of abortions, preterm births, other reproductive effects, fecundity, hormones, inconsistent. Animal studies. Show anti-adrenergic activity, PP Heinz DDD, estrogenic activity, PPDBDD. Male exposure decreases reproductive tissue weights. Female exposure increases uterine weight, decreases fertility estradiol. Mild thyroid dysfunction seen in animals, less clear in humans. Timing of exposure is crucial. Might explain inconsistencies. Developmental exposure impacts health later in life. Relevant today for older adults born before ban links to increased risk of type 2 diabetes, obesity, liver cancer later. Shows how early exposures cast long shadows.
The everywhere plasticizers.
Phthalates seem to be in everything. Plastic containers, personal care products, medical tubing. What makes them so pervasive and what are their effects?
Phthalates are used as plasticizers, making PVC flexible, durable, clear. Used in flooring, wall coverings, medical devices, food packaging, cosmetics, fragrances, pharmaceuticals. Prevalence plus plastic waste it views as unavoidable exposure. Not chemically bound, leach out easily.
Exposure routes.
Ingestion, inhalation, germal absorption. High levels like DHP in meats, oils, dairy, packaging, processing. Food wrapped in plastic. Air, water, dust near manufacturers. Crucially, cross-placental barrier, fetal exposure.
Chemistry and mode of action.
Esters of phthalic acid.
Yeah.
Grouped by chain length. Long chain, high MW like DHP, DMP plasticizers. And short chain, low MW like DMP, DEP personal care. EDC activity via multiple mechanisms. Modulate hormone levels, alter receptor expression, direct receptor binding, epigenetic modifications. Rodent studies show non-monotonic effects. Higher doses sometimes have opposite effects to lower doses. Makes risk assessment complex. Directly bind nuclear receptors, NRs mimicking steroid ring. Influence AR, ER, PPR signaling, activate or inhibit depending on phthalate NR co-activators. Also affect converging pathways, MAPK, NFK, PI3, ACT, alter cell proliferation, apoptosis. Depends on route. Hydrophobicity. Urometabolites, similar across threads. Dermal absorption 160%, oral, higher. First pass metabolism, diesters, monoesters. Low MW monoesters excreted in urine. High MW undergo phase 2 conjugation first. 60% eliminated urine and feces after dermal, 80% after oral. Rapidly metabolized plasma half-Liz 24Hers. Measure urine metabolites for recent exposure.
Health effects. Developing systems seem key.
Yes, developing reproductive system is particularly sensitive target. Human-animal studies. Adults linked to reduced reproductive ability, lower semen quality, increased pregnancy loss, increased risk of type 2 diabetes, obesity, inflammation. Children associated with decreased LH testosterone, reduced anogenital distance, male infants, thyroid dysfunction, precocious puberty. Bisphenol A, BPA, the ubiquitous plastic component.
BPA, another one with lots of public attention. BPA-free everywhere. What's the science? Is it still a concern? Are replacements better?
Bisphenol A, BPA, is prevalent, uses plasticizer in hard, clear polycarbonate plastics and epoxy resins, protective coatings. Primary route. Ingestion from food beverage packaging, water bottles, can linings, bottle tops. Indirectly from contaminated water food via leaching. Leaching increases with heat, non-neutral pH, detergents, physical disruption. Crosses placenta, found in cord blood, amniotic fluid, fetal plasma. Also in breast milk, infant exposure.
Chemistry and mode of action. Estrogen-like.
Structurally similar to estrogen, binds ERI and EUA, weak estrogenic activity, 1000x lower affinity than estradiol. Can activate ERI, but differentially interacts with ERA, prevents proper signaling. ERI acts as selective ER modulator, agonist, antagonist. Most studies focus on high-dose transcriptional effects, but low concentrations can interfere with non-genomic ER pathways, rapid signaling, TR2 plus release.
ADME, how does the body handle it?
Significant first-pass metabolism, intestines, liver, via CYP2C enzymes, glucuronidation, sulfation. BPA monoglucuronide lacks estrogenic activity, is main metabolite in bilirubin. Body tries to detoxify.
Health effects, found in almost everyone.
Yes. Identified in urine of nearly all adult children, also pregnant women's serum, breast milk, amniotic fluid, cord blood, placental tissue. Ubiquitous.
So most of us have BPA right now. What are the observed health effects? How certain are we?
Despite challenges proving causation in human studies, evidence supports BPA is harmful to human endocrine system, fertility, sexual function, higher urinary BPA associated with lower serum E2, poor oocyte yield maturation, higher implantation failure, IVF, decreased male sexual function, lower sperm production count motility, women of childbearing age, higher BPA linked to shorter luteal phase. Urinary BPA also correlates with type 2 diabetes, thyroid dysfunction. But human studies often have limitations, small size, confounders, design issues, association, not always causation. This inconclusiveness leads to differing regulations globally, though bans in baby bottles common. Consumer demand led to BPA-free labels. But replacement chemicals, BPS, BPF, often less studied concern about regrettable substitutions.
What about lab studies?
Provide more direct evidence. Mice exposed to BPA, impaired reproductive organ sperm development, altered testosterone, alters brain function, neurohormone receptors, thyroid hormone gene expression, offspring of exposed dams, decreased insulin secretion. BPA has greatest effect during critical developmental stages, embryonic, gestational, neonatal, childhood, puberty. Why should you care? Because even low-level exposures during these windows can have lifelong consequences. Makes informed choices about consumer products relevant for long-term health. 7. The immune system under attack. Immunotoxicants.
We've covered organs, hormones. What about the immune system, our body's defense? How do chemicals mess with that?
Crucial area. Immunotoxicants are xenobiotics that interfere with modulate immune function. Several mechanisms. Functional changes. Modulate immune cell abilities, antibody production, cytokine release, antigen presentation, proliferation differentiation. Structural changes, alter cell receptor expression, damage lymphoid organ architecture, thymus, spleen. Compositional changes, alter proportions numbers of immune cell populations. And since the immune system interacts with other organs, chemicals like EDCs can indirectly impair immune function. Disruption leads to either immunosuppression, increased risk of infection, cancer, or immune overactivation. Increased risk of autoimmunity, allergy, hypersensitivity. Both extremes are bad.
The potent immunotoxicant?
TCDD. Notorious history. Agent Orange, Seveso. What makes it such a potent immunotoxicant? How are we exposed?
TCDD, 2,3-hongol-7-cos tetrachlorobenzo-p-dioxin, is a byproduct contaminant formed making 2-fulver-8, 4-ether-5, trichlorphenol, herbicide, fungicide, component of Agent Orange, combustion product, burning organic matter, persistent pollutant, resists breakdown, notoriety from Agent Orange, Seveso accident, Italy, 1976, assassination attempt on Viktor Yushchenko, Ukraine, 2004, extremely persistent.
Those are today.
Lipophilic, accumulates in food chain, 90% exposure today from ingesting contaminated fat-rich foods, dairy, meat, fish, occupational exposure from waste incineration, natural combustion.
Chemistry and mode of action.
Dioxin family, 400 chemicals. TCDD, most potent, high affinity for aryl hydrocarbon receptor, IHR, resists metabolism, strong, sustained AHR activation. Immunotoxicity mediated via IHR, expressed in most immune cells. T cells' primary target, promotes differentiation towards regulatory C cells, influences antiviral CD8 plus T cells, induces tolerogenic dendritic cells, inhibits B cell antibody production. Oral exposure, 90% absorbed in oil, 50-60% diet. Distributes widely, bioaccumulates in liver adipose tissue, lipophilic. Resistant to metabolism, human half-life, 7-11 years. Elimination mainly feces, very slow. Extreme persistence. Health effects. Effects mainly from occupational and accidental exposures. Consistent findings. Chloracne, increased risk of certain cancers. Immunological impacts. Consistent reduction in CD4 plus T helper cells, humans, primates. Animal models. Thymic involution atrophy. Rodents more susceptible to infections. Influenza, trichinella. Interestingly, it doesn't increase susceptibility to all infections, can even be protective sometimes, dampens inflammation. Highlights complex, context-dependent roles of AHR, ligand, dose, route, tissue, timing matter. IHR, ligands, and particulate matter, PM, the air we breathe.
So AJ is central. How does it relate to something widespread like air pollution, particulate matter? It seems different.
Perceptive connection. RHR signaling is critical for immune cell development maintenance function. Absence lack of ligands disrupts homeostasis. Key roles in Th17 differentiation, innate lymphocyte development, IL-22 production. Pro-inflammatory effects of IHR activation evident with air pollution ambient PM.
Air pollution, huge health issue.
Major trigger exacerbator of asthma, allergies, COPD, increases risk of cancer, autoimmune diseases. WHO estimates millions of premature deaths globally each year. PM consequences depend on physical properties, size, shape, and absorbed chemicals. PAHs, PCBs, metals. PAH, dioxin-like PCBs on PM activate AHR in vivo ex vivo, inducing CYPs. Unlike TCDD's systemic immunosuppression, IHR activation by PM linked to increased RS-oxidative stress, interactions with NF-PBNRF2, inflammation antioxidant response, increased inflammation. PM 2.5 specifically alters dendritic cells towards Th17, promoting phenotype, AHR-dependent. Pro-inflammatory outcomes seen in Seveso cohort, exposed individuals 2x likely to get COPD. Highly relevant given overlaps with COVID-19, wildfires, climate change impacting air quality.
The forever chemicals.
Forever chemical striking nickname. Why are these per and polyfluoroalkyl substances so persistent? What are their impacts?
PFAS are anthropogenic, widely used in firefighting foams, commercial surfactants, dubbed forever chemicals due to exceptionally long half-life, environmental, biological. Carbon-fluorine bond, incredibly strong, resistant to degradation.
Exposure. Where are they?
Hydrophobic, oleophobic properties used in food packaging, greaseproof paper, cookware, furniture, carpeting, clothing. Exposure via consumer use, contaminated drinking water, air, airports, military bases, chemical plants, inhalation of indoor dust, dermal, less significant, don't degrade, accumulating since 1940s. Legacy PFAS, PFOS, PFOA, phased out voluntarily in U.S., still used globally. New, next-gen, shorter-chain replacements continuously developed. 4,000 PFAS synthesized, 100 measured in environment. Complex, pervasive issue.
Chemistry and mode of action.
Contain O1 perfluoroalkylated, fully fluorinated carbon chain, plus functional head group. Properties vary with chain length head group. Long chain, C7 or S6. Bioaccumulate more. PFO, APFOS, most studied, interfere with endocrine function, lipid metabolism, steroid biosynthesis, induce ER stress, ROS, directly interfere with PPRR, NRF2, NFL pathways,
ADM.
ingestion, 90% absorbed. Partition to high protein matrices, bind serum albumin, PFOA, PFOS, favor liver, next-gen PFH, GOSES, favor serum, highly resistant to metabolism, bioaccumulate, primary elimination, bile, urine, menstruation, lactation, additional routes, maternal transfer, serum levels higher in males, partly due to menstruation elimination in females. Human half-lives PFOA, PFOF, 5 years, reinforces forever nature.
Health effects. Immune system.
Based on epidemiology animal studies, PFOS, PFOA presumed immune hazard to humans, largely based on studies showing negative correlation between PSAS levels and antibody responses, vaccine effectiveness measure. What's particularly concerning, several studies show developmental exposures in utero-early childhood correlated with reduced antibody response to childhood vaccines. Levels for tetanus diphtheria fell below protective levels in some exposed Greenlandic children. Supported by murine studies, PFAS suppresses T-cell-dependent antibody response. Robust immunosuppression assay. Also evidence, human mouse, that PFAS can increase hypersensitivity responses, allergies, autoimmunity, though data inconsistent, depends on dose, timing, duration, understanding mechanisms crucial for future studies. 8. The gut's hidden role, microbiome and environmental toxicants.
We've covered organs, hormones, immune system. What about the tiny world inside us, our gut microbiome? Trillions of microbes. How do toxicants affect them? Is it one-way harm, or can microbes help?
Rapidly emerging, incredibly important area. Exposure to toxicants can cause significant structural functional changes in the gut microbiome. Consequences of gut microbiome toxicity, loss of bacterial diversity, critical for resilience, changes in bacterial metabolites, crucial signaling molecules, alterations in energy metabolism balance, loss of diversity increases susceptibility to pathogens, altered metabolites impact host physiology homeostasis widely, disturbed energy metabolism linked to obese nutrition.
So a healthy gut is another defense line, but also a target, a two-way street.
Exactly. Dysbiosis, an imbalance caused by chemicals linked to many diseases. IBD, obesity, diabetes, cardiovascular liver disease, colorectal cancer, even neurological disorders, gut-brain axis. Pesticides can disturb bacterial communication, quorum sensing. Heavy metals directly change microbiome composition, alter metabolic capabilities. And here's the fascinating two-way street. The gut microbiota can also mediate the toxicity of environmental chemicals. They influence whether a chemical becomes more or less toxic. Gut flora express enzymes that can bioactivate, make more toxic, or detoxify compounds, key enzymes. Azoreductases, nitroreductases, glucurondases, sulfatases, meliases. These metabolize diverse chemicals, PAH, nitrotoluenes, pesticides, PCBs, metals, azodyes, melamine, artificial sweeteners. Raises an important question. Our gut is a crucial interface, adding complexity. The balance of our microbes might determine our vulnerability or resilience to environmental threats. Deeply interconnected. Outro.
What an incredible deep dive we've had today into the complex, often unseen world of environmental and occupational toxicology. From the nuanced dance of risk assessment, epidemiology, toxicology, to the specific, sometimes insidious ways metals like lead, mercury, arsenic, cadmium, chromium interact with our biology, the pervasive disruption by endocrine disruptors like DDT, phthalates, BPA, the subtle attacks by immunotoxicants like TCDD and PFAS, and even the surprising, crucial role of our gut. It's clear our daily environment has profound, often unseen impacts.
We've truly traversed the landscape, haven't we? From macro public health challenges to intricate molecular mechanisms, understanding these concepts, the precautionary principle, specific chelators, intergistic effects like aflatoxin and Hep B, it provides a powerful framework for being a well-informed citizen. You now have a comprehensive understanding on par with elite professionals in this field.
Absolutely. You now possess that comprehensive understanding. And armed with this knowledge, you can approach the world with a much more informed perspective, recognizing the silent threats and the complex interplay affecting your health every single day. This isn't just theory, it's about what's happening in your body, your environment.
Indeed. Knowledge is most valuable when understood and applied. And as we conclude this deep dive, consider this provocative thought. Given the invisible and long-term impacts of many of these environmental chemicals and the profound, subtle ways they interact with our bodies, what responsibility do we, as increasingly informed citizens, have to advocate for continued research, stricter regulations, and innovative solutions to ensure healthier environments and thus healthier lives for ourselves and for future generations? The answers lie not just in science, but in collective action and informed stewardship.